So I want you to imagine this incredible, highly trained molecular assassin.
Right.
We're talking about an advanced antibody drug, engineered in a lab, and it is specifically designed to seek out and destroy a cancer cell.
The ultimate weapon, basically.
Exactly. It sounds like the perfect weapon.
Yeah.
But there is a really glaring problem here. When these assassins are actually deployed into solid tumors in the human body, they fail to trigger a response like 80% of the time.
Yeah. And I mean, it's one of the most persistent hurdles in modern oncology today. You have this therapy that neutralizes cancer perfectly in a controlled petri dish, right?
Yeah.
But the moment you put it into a living, breathing patient with a solid tumor, the drug suddenly just doesn't do the job.
And the question driving cancer research has basically always been why? I mean, do these molecular assassins get lost in the bloodstream?
Hmm.
Do the cancer cells somehow disguise themselves?
Or is there a physical barrier? Yeah.
Right. Is there a literal physical wall stopping the drug from ever reaching its target? So welcome to this deep dive into the source material. Today, we are exploring a genuinely groundbreaking paper published in Nature Biotechnology back in June 2026. And it finally gives us a map of this microscopic battlefield.
It really does.
We're looking at a revolutionary framework called Single-Cell Spatial Pharmacobiology, or SSP. It is a completely new way to track exactly where a drug goes, cell by cell, inside a living human tumor.
And to really understand how they did this, we need to look at the data set first.
Okay, lay it out for us.
So this study draws on phase 1 clinical trials that were conducted at the Stanford Cancer Institute between 2018 and 2021. The researchers analyzed 50 participants who had one of 3 specific types of solid tumors.
Right.
We're looking at head and neck squamous cell carcinoma, which we'll just refer to as HNSCC.
That makes sense.
Then there's pancreatic ductal adenocarcinoma, or PDA, and non-small cell lung cancer, or NSCLC.
Got it. But, tracking a single drug molecule's journey through the absolute chaos of a human tumor, that just seems almost impossible to me.
Yeah.
I mean, for a long time, the tools we had were just incredibly blunt, right?
Oh, very blunt. Historically, I mean, we've relied really heavily on blood tests, which, they only tell you how much of the drug is circulating in the patient's bloodstream.
Not where it actually ends up.
Exactly, not where it ultimately ends up in the tissue. Or, doctors use full-body PET scans, and a PET scan gives you a great organ-level picture. You might see a glowing area where the tumor is...
What's blurry?
Right. It completely lacks the cellular resolution to tell you if the drug is actually binding to the cancer cells or if it's just, pooling in the dead space right next to them.
Wow, okay.
What makes SSP so revolutionary is that it finally provides that cellular resolution. It shows us the exact neighborhood, the specific cell, and the actual molecular interaction.
So before we get into the reasons why the drugs are failing, we really need to understand how the scientists managed to track them at that impossibly small scale. I mean, building a microscopic GPS kind of seems like science fiction.
It starts with the drug itself, actually. The researchers used a well-known antibody drug called panitumumab. Its specific job is to hunt down and bind to a protein on the surface of cancer cells called EGFR. But they didn't just infuse the standard drug. They chemically attached a fluorescent near-infrared dye to it.
Oh, that's clever.
Yeah, and this modified version is called PAN-800. So they created a drug that literally glows under specific microscopic imaging.
Okay, making the drug glow makes total sense. But if it's just glowing in a massive sea of tissue, there's still the issue of dosing, right?
Yeah.
Like if you give a patient a massive tumor-destroying dose of this glowing drug, wouldn't it just flood the entire area and make it impossible to see where the natural roadblocks are?
That is exactly the problem with tracking a standard therapeutic dose. If you blast a tumor with a massive saturating amount of a drug, the sheer pressure and volume might just force it everywhere.
Right, it would hide the bottlenecks.
Exactly. It masks the tumor's natural defenses. So to get around this, the patients in this study received what's known as a subtherapeutic dose.
Ah, a much smaller amount.
Right. They were given this glowing PAN-800 drug purely for surgical guidance, just days before their standard of care surgery to have the tumor removed.
So the original goal was just to help the surgeon see the glowing edges of the cancer in the operating room to ensure they got it all out.
Yeah, exactly. But for the researchers who were analyzing the tissue afterward, this low dose was the key to everything.
Because it wasn't overwhelming the system.
Exactly. By using a trickle instead of a fire hose, they could observe the natural gradients of the drug. They could trace exactly where the fluid easily flows and precisely where it hits a wall and just stops dead in its tracks.
Man, that's brilliant. But just seeing a glowing drug stop at an invisible wall isn't quite enough, is it? You need to know what that wall is made of. Like, if the drug is the GPS signal, you still need a map of the city.
Right, and that map was created using a technology called codex. In clinical terms, codex is highly multiplexed spatial proteomics.
Okay, that's a mouthful.
It is. Basically, once the tumor was surgically removed, they sliced it incredibly thin. Then they applied DNA barcoded antibodies to the tissue slice.
Okay, let's break that down for a second. Spatial proteomics essentially means they aren't just putting the tissue in a blender to see what proteins are inside, right?
Right.
They're keeping the tissue totally intact to see exactly where those proteins are physically located in space.
Precisely. They map dozens of different proteins simultaneously, and this allows them to identify and color code every single tumor cell, every immune cell, every blood vessel, and every structural fiber in the tissue.
Wow.
And they do all of this on the exact same slide where they're already tracking the glowing PAN-800 drug.
That is wild. It's like looking at a satellite image of a city at night. A traditional PET scan just shows a blurry, glowing area, and you assume the drug was delivered.
Yeah.
But combining the glowing drug with this codex map, it's like having a GPS tracker on the individual mail carrier delivering the drug, overlaid on a three-dimensional blueprint of every single street, every barricade, and every individual house in that city.
That's a perfect analogy. And because you have both the drug's exact location and the entire architectural context surrounding it, you can finally see who is receiving the package and who is just being skipped.
Which really brings up an interesting thought. If you're looking at this incredibly detailed map of a tumor city, you have to wonder what the biggest physical obstacle actually is.
I
mean, is the city itself built differently depending on the type of cancer?
It absolutely is, and that was the first major finding when they zoomed out to look at the macro view. They compared these different tumor environments, the pancreatic cancer, the non-small cell lung cancer, and the head and neck cancer. And the drug behaved radically differently depending on the city it was in, so to speak.
Yeah, the data on the bulk tissue is really striking. When they looked at the overall drug concentration, the pancreatic tumors, the PDAC, had significantly lower levels of the drug compared to the head and neck and the lung cancers.
Well, pancreatic cancer is notorious for something called a desmoplastic reaction.
What's that?
It essentially produces a highly dense fibrotic stroma. If we stick with your city analogy, a PDA tumor is like a city made entirely of thick concrete bunkers with virtually no open roads.
Oh, wow.
Yeah, it's an incredibly hostile, dense physical mass that the drug simply cannot penetrate.
So PDAC is universally tough, but the lung and the head and neck cancers generally allowed more of the drug inside.
Generally, yes.
However, when you look specifically at the head and neck cancers, the inter-participant heterogeneity is massive. The difference from one patient to the next is staggering.
It really is.
Like, some patients had drug concentrations as low as 6.4% of the injected dose per kilogram, while others absorbed up to 38.1%.
And that variance is the core of the problem.
Wait, hold on. I need to make sense of that math. If I weigh the exact same as another patient with the same type of head and neck cancer, and we both get the exact same dose calibrated from the IV bag,
Right.
how on earth is my tumor absorbing 6% and theirs is taking in nearly 40%? That shouldn't even be possible if delivery is just based on blood flow and dosage.
It's a fundamental shift in how we have to think about oncology. The problem isn't the dosage circulating in the blood.
Right.
The bottleneck is the specific, localized architecture of that individual patient's tumor.
Okay, the actual structure of it.
Yes. Even within the exact same type of head and neck cancer, one patient's tumor might be highly vascularized with plenty of open pathways and blood vessels, while another's might be actively building defensive walls. The success of the drug delivery is dictated almost entirely by the tumor's unique geography.
So the pancreatic cancer acts like a concrete bunker blocking the drug across the board. But the head and neck cancer is wildly unpredictable.
Exactly.
It really proves that getting the medicine into the tumor depends on that unique architectural landscape.
Yeah.
But if the total amount of drug getting into the tumor varies so drastically, what actually happens when we zoom all the way in? Like, what happens when we look at the specific individual cells to see if they are actually receiving the medicine?
That transition to the microview is where the study gets incredibly revealing. The researchers needed to see if the cancer cells themselves were somehow playing a role in this uneven distribution.
And to do that, they had to measure target engagement.
Right.
Because it's not enough for the drug to just float by the cell. It has to actually plug into the target receptor to do its job.
Exactly right. The target for panitumumab is the EGFR protein.
Okay.
When the EGFR pathway is active and telling the cancer cell to grow, a specific version of the protein called phosphorylated EGFR, or PEGFR, is present.
So it's like a switch.
You can think of PEGFR as the on switch, yeah. And the researchers found a direct inverse correlation. In the specific microscopic areas where the pan-800 drug delivery was high, PEG4 was low.
So the drug was successfully binding to the receptors and flipping that switch to off, shutting down the cancer's signaling pathway.
Yes. The molecular assassin works perfectly if it can get there.
Getting there seems to be the entire problem, doesn't it? I mean, the researchers analyzed a staggering 1 million individual segmented cells across these patients. And out of all the tumor cells, only 16.6% were both EGFR positive, meaning they had the target, and pan-800 positive, meaning they actually received the drug. Only 16.6%.
Yeah, but the truly concerning statistic is the other side of that coin. 35.9% of the tumor cells were EGFR positive, but pan-800 negative.
Oh, man.
Over a third of the cancer cells had the exact target the drug was hunting for, but the drug never reached them. They were completely untouched by the therapy.
That's crazy. If the drug is actively floating around inside the tumor tissue, why are more than a third of the completely vulnerable cells just being ignored?
Well, when the researchers mapped the exact spatial coordinates of those specific cells, they discovered a phenomenon known as the binding site barrier.
The binding site barrier. Okay.
It all comes down to their geographic layout. The cells that successfully grabbed the drug, that 16.6%, they were overwhelmingly located at the extreme outer edges of the tumor nests, right next to the blood vessels where the drug initially spills out into the tissue.
Okay, I see where this is going.
Yeah, and the 35.9% that were starving for the drug, they were trapped deep inside the inner core of those tumor nests.
It almost sounds like a buffet line.
A buffet line.
Yeah, imagine the tumor nest is a large room, right? And the blood vessel is the door where the buffet cart arrives. The tumor cells on the outer edge are the greedy people standing right at the front of the line.
Yeah, I see.
Because this antibody drug binds so tightly to its target, and because the dose is relatively low, that front row of cells just piles their plates high. They absorb absolutely all of the medicine immediately.
That is a remarkably accurate way to put it.
And by the time the cart tries to push toward the back of the room, there is literally nothing left for the tumor cells hiding in the core. So they just starve for the drug.
Exactly. The high binding affinity of the drug essentially turns that outer layer of cancer cells into a sponge. They become their own barricade.
Wow.
By greedily absorbing all the therapeutic molecules, they physically starve the inner core cells of the medicine they need, protecting the rest of the tumor from the treatment.
So even when a drug is perfectly engineered, only about 16% of the cells might actually get it because the tumor cells act as their own barricade.
Right.
But cancer cells don't just float in a vacuum. They live in a physical environment, a neighborhood. And if the cancer cells are forming the first barricade, you really have to wonder what the rest of the neighborhood is doing.
Which brings us to what might be the most groundbreaking part of the entire study.
Okay, let's hear it.
It isn't just about how the tumor cells behave. It's about the infrastructure they actively build around themselves. We're talking about the stroma, or the connective tissue, and specifically the extracellular matrix.
The ECM, right. Historically, when drugs failed to penetrate solid tumors, scientists usually pointed the finger at collagen.
Right, collagen.
Because it's a very common structural protein in the body, so it's just the most obvious suspect getting in the way.
But the high-resolution codex mapping revealed a completely new primary culprit. It wasn't just generic collagen causing the issue. The primary physical barrier limiting the diffusion of the antibody drug was a specific protein network called periostin.
Periostin.
Yes. They observed that periostin-rich ECM networks were forming these dense, highly organized physical walls that were literally choking off the drug's movement through the tissue.
But periostin doesn't just magically arrange itself into a wall, right? Some biological mechanism has to build it. And this is where the biology gets genuinely sinister, in my opinion. The tumor recruits local cells called FAP plus CFs.
Yeah, CIF stands for cancer-associated fibroblast. And to understand why this is so critical, you have to look at what normal fibroblasts usually do.
Okay, what do they do?
In a healthy body, fibroblasts are essentially the medics. They are responsible for creating connective tissue and rushing in to aid in wound healing by building scar tissue.
Okay, so they're the good guys.
Usually, yes. But FAP plus CF, which stands for fibroblast-activation-protein-positive, is a mutated, corrupted version. The tumor has essentially hijacked these local healing cells and reprogrammed them.
So it tricks them into thinking the tumor is a wound that needs to be protected and scarred over.
Yes.
That means the tumor isn't just passively sitting there hoping the immune system or the drugs don't reach it. It has actively drafted these FAP plus CK construction workers to build a custom periostin bunker around itself.
It is an active dual-threat defense system. The FAP plus CCAFs are the enemy engineers, and the periostin is the quick-drying concrete they continuously pour to create this massive stromal barrier.
Man, that is terrifying. And the researchers didn't just guess this based on seeing proteins near each other, did they? They actually validated the entire mechanism using another layer of cutting-edge technology.
Right. To be absolutely certain that the CCAFs were the ones building the wall, they used an orthogonal method called xenium spatial transcriptomics.
And just to clarify the difference there for everyone, if Codex is looking at the finished proteins like the building materials, Xenium is looking at the RNA, which are the genetic blueprints the cells are actively reading to make those materials.
Exactly. They looked at the exact same tumor tissues and found a perfect inverse correlation.
Okay.
In the specific areas where the FAP plus CCAFs were highly expressing, the POSTN gene, which is the genetic blueprint that creates periostin drug delivery, was completely choked off. The cells were actively reading the blueprint, pouring the concrete and stopping the drug, all in the exact same microscopic location. And what makes this so significant is that this specific barrier was conserved across both the head and neck cancers and the pancreatic cancers.
So the extracellular matrix isn't just passive background tissue. It contains these dense walls of periostin, and the tumor actively hijacks local FAP plus CCAFs to act as construction workers, constantly pouring this concrete to protect the cancer.
That's exactly what's happening.
Knowing exactly what the wall is made of and exactly who built it, I mean, that fundamentally changes the way we have to think about fighting cancer.
It forces the entire oncology field to synthesize all these findings and look at the larger picture. This level of single-cell spatial pharmacobiology proves that for solid tumors, the era of simply designing a sharper molecular assassin in a vacuum, and just hoping for the best, is over.
Because the efficacy of a drug is no longer just about whether the molecular key fits perfectly into the lock on the cancer cell. It's about whether that key can navigate a chaotic three-dimensional maze. It has to get past the greedy cells at the front of the buffet line, and it has to somehow breach those dense periostin walls being actively built by the hijacked FAP plus CCAFs.
And if that wasn't difficult enough, there is one more fascinating side note from the study that highlights just how hostile this tumor microenvironment really is.
Oh, there's more.
Yeah. The researchers also caught certain immune cells, specifically macrophages and myeloid cells, literally gobbling up the PAN-800 drug.
You're kidding. So they act like molecular thieves.
Yes, exactly.
So you have a drug navigating a maze, trying to break through concrete walls. And on top of all of that, you have local immune cells essentially mugging the mail carrier and stealing the drug before it can ever reach the cancer cell.
It is a profoundly hostile environment on every conceivable level, but the true power of SSP is that we no longer have to guess why a therapy failed.
You can finally see it.
Exactly. We can finally see all of this happening in a living human tumor. We can map the failures to engineer better successes.
Which leaves a really provocative thought to mull over, I think. If we now know with absolute certainty that these hijacked FAP plus CFs and their thick periostin walls are the primary physical barriers stopping life-saving drugs from reaching the tumor, what if the next generation of cancer treatments aren't designed to kill cancer cells at all?
Oh, that completely reframes the objective.
Exactly. What if our next great medical breakthrough isn't an assassin, but a bunker buster drug?
A bunker buster, I like that.
Right. Imagine a therapy that ignores the cancer entirely and instead targets only the architects and their concrete. A drug designed specifically to wipe out the FAP plus CFs and dissolve the periostin, effectively clearing the road.
That makes total sense.
Once the barriers are gone, the patient's own immune system, or even standard older therapies, could simply walk right in and finish the job. So think about that the next time you hear about a new cancer trial. The molecular assassin isn't failing because it's weak, it's failing because we haven't given it a clear shot.
That is a really fascinating perspective. I mean, it changes the entire paradigm of drug delivery in solid tumors from a brute force approach to like a strategic demolition.
It really does. So the next time you think about how we target disease, keep that image of the molecular assassin and the concrete bunker in mind. The map has finally changed. Until next time.